Hello everyone, I am Fashwa Farooqi and you're watching Fashwa Farooqi YouTube channel. Okay, the topic for our today's video is DNA replication. We have discussed about the DNA replication and the experiment performed by the Michelson and Stahl in my previous video. If you want to know about this experiment performed by Michelson and Stahl, go and check out my previous video. Now let's just get started. What are we going to learn in this video? Firstly, we will discuss about the definition of DNA replication, and secondly we will discuss about the mechanism of DNA replication completely. Okay, get started. Definition of DNA replication. What is DNA? Firstly, I have told you many times in all my previous video, the DNA is double-stranded helical structure containing three basic components. The first one is a nitrogenous base, second one is a pentose sugar, and the third one is phosphate group. Now what is replication? The replication means formation of formation of exact copies. Okay, the process by which DNA of living organisms give rise to a copy of itself is called DNA replication. It's a definition of DNA replication, that DNA will give rise to its exact copies. Okay, now let's move toward the mechanism. Firstly, we should know about the enzymes that are involved in the DNA replication. These are some enzymes that are involved in DNA replication. The first one is helicase. The second, we will study the roles of all these enzymes step by steps. Okay, now let's just know the names of these enzymes. Helicase, the second one is DNA binding proteins, or these are also called as single strand binding proteins. Okay, the third one is primase. We will study its function also. The fourth one is DNA polymerase 1, DNA polymerase 2, DNA polymerase 3, and the final is ligase. Now, before studying the mechanism, let me tell you about the origin of replication. What is origin of replication? The DNA, in the DNA double helicase, there are certain points that from where the replication starts. That point is called as origin of replication, and there are many origin of replications in one DNA molecule. Okay, so this is my DNA molecule, and this is the, suppose it is the replication origin containing a specific kind of specific type of nucleotides in it. Now the replication will begin from this point. Okay, so it is the replication, origin of replication. Okay, now let's study about the first enzyme and its role. The first enzyme we have is helicase. What is its role? Helicase, basically what does it do? It works as a unzipping enzyme, basically, which will unzip or separates the two parental DNA strands. Okay, the helicase breaks down the hydrogen bonds between the bases. Which bases are these? These are nitrogenous bases. As you know that the DNA contains nitrogenous bases, adenine, guanine, cytosine, and thymine, and those all bases are connected to each other by this hydrogen bond. So that's why the DNA forms a double helical structure. These are, you can say, these are nitrogenous bases, and these are connected to each other by hydrogen bonds. Now the helicase, what will helicase do? It will just breaks down all these hydrogen bonds so that the two strands may unwind. Okay, you can see this is helicase working here, and it will work as unbinding or unzipping enzyme that will just breaks down hydrogen bonds between the nitrogenous bases so that the two strands of the parent may separate. Now after separation we will get a replication fork. You can see this is a Y-shaped structure. The two parental strands, when the two parental strands are separated, they form a replication fork. This is a replication fork, or it is also called replication bubble. You can see a bubble here. This is replication bubble, and this point is called replication fork. Okay, now let me revise it again so that it may be clear to you. Helicase, it works as a breaking down, basically works as unzipping enzyme, which will breaks down the hydrogen bonds present between the nitrogenous bases. Okay, and will form, after breaking those hydrogen bonds between the nitrogenous bases, what will the result, what will be the result we get is we will get two parental strands forming a fork, replication fork. Okay, this is helicase, and these are two parental strands of DNA that are forming replication fork. Okay, now let's study about the next enzyme we have, single strand binding protein. Now this or I have told single SSBP, we also in short form we also called it as SSBP. Okay, what will these two, this second enzyme will do? It will basically clip both the strands so that they may not connect each other again. These are the two parental strands of DNA that were separated by helicase. Now what is the role of single stranded binding protein? They will hold the single stranded so that they may not connect each again, so that the hydrogen bonds may not form again between them, and again the double helical structure of DNA may not form. Okay, so it basically clips the single strands of DNA so that they may not connect again, basically and they just remain far apart from each other. And the third enzyme we have is primase. Primase, what does primase do? Primase will form RNA primer. And what is RNA primer? RNA primer is basically the sequence of 10 RNA nucleotides. What is the role of, why do we need RNA primer? To start the replication before forming a new strand on the single strand of parental molecule. Now in this case you can see that we have single separate parental strand, and a new molecule, new strand of DNA will form on this parental molecule, and a new strand of DNA molecule will form on this parental molecule, so that we will get two molecules of DNA. Okay, so the replication will basically need primase to form a primer. Primer is basically RNA primer, which contains 10 RNA nucleotide sequences. Here in this case you can see this is primer, which is red in shade, and in this it is a leading strand. It contains just one primer, because in this case our replication is continuous, while on lagging strand you can see there are multiple primase. This is also a primase, and this is also a primase. Multiple primase we have, because in lagging strand the replication is not continuous. We just have segments of DNA molecule will form on lagging strand. Okay, next, DNA polymerase 3. It is the most important enzyme in the DNA replication, as it makes the new strand of DNA molecule. Here in this case you can see the red strand is the new strand, and the blue strand is the parental strand, which acts as a template over which new strand will form. Okay, now DNA polymerase, what does it do is DNA polymerase will form new strand of DNA by adding new nucleotides, and it will always start its work from the RNA polymerase. It cannot synthesize a new strand of DNA without RNA primer. As you know that in our previous step we had made our primer, RNA primer, which was formed by the primase. The DNA polymerase 3 cannot start the formation of new strand without primer. This is primer, and here the new strand is formed by the DNA polymerase 3. This green you can see. Okay, now another thing is the most important thing, that it can form new strand along 5 to 3 direction only. As you know that the two strands of the DNA are anti-parallel to each other. The first strand here, in this case the parental strand, is from this is 3' end, so the new strand of the DNA will form from 5 end and will move towards the replication fork, and this end will be 3' end. So this is a simple, the leading strand is a simple one, because it just starts from 5' end and will ends towards 3 end. Now what about this strand? It cannot start from 5 end here, because this is also a 5 end, so it cannot start from 5 end. Here in this case it will start from this point. Okay, now we will know about this more about the leading strand and the lagging strand in our next slide. Wait a sec. Now here in this case you can see our leading strand. First we will study about the leading strand. Now what is leading strand? The new strand formed that is elongated towards the replication fork. Firstly, what is replication fork? I have told you this point is a replication fork, which is Y-shaped. As you can see, it's Y-shaped, and this is replication fork, and this is our leading strand. It is moving towards the new strand which is forming, is moving towards the replication fork here. Okay, so and its growth is continuous. You can see that the growth is continuous. No fragments are formed, no segments of new strand are formed. The growth of new strand is continuous. The new nucleotides are being formed and continuously being placed according to their sequence. Okay, now before studying about the lagging strand, you should know about the sequence and the nucleotides. As you know that what is nucleotide? Nucleotide is made up of 3 basic components. Okay, the DNA polymerase 3 uses or synthesize these nucleotides and then arrange the nucleotides according to their sequence to form a new strand of the DNA. This nucleotide, what does it contain? It contains 3 components. First is 5-carbon sugar, the second is nitrogenous base, and here the third component is phosphate group. Now let's know about the position. Here the carbon number 1, this is the carbon number 1, and at this point nitrogenous base is attached, and the second important point is third carbon. Here you can see the carbon number 3, this is carbon number 3, and to this carbon hydroxyl group is attached. This is OH group. It is important, it will play important role in the formation of new strand. This hydroxyl group and the phosphate group which is attached to carbon 5. Okay, these two things, the phosphate group attached to carbon number 5 and the hydroxyl group attached to carbon number 3 will play the most important role in the formation of new strand. How will these nucleotides will connect, and how does the new strand will form? Here in this case you can see the phosphate group This is phosphate group attached to the carbon number 5 of this nucleotide. Here we have two nucleotides. This is first nucleotide and this is second nucleotide. I have shown that how does these nucleotides are connected to each other so that they form a chain. Here you can see the phosphate group attached to carbon number 5 of this nucleotide, and this is hydroxyl group which is free. OH group, this is free, it is not attached to any nucleotide, and this is phosphate group of first nucleotide attached to carbon 5, and here in this case you can see the hydroxyl group of this nucleotide attached to carbon 3 is forming a bond with phosphate group of the first nucleotide. So in this case this is called a phosphodiester bond, which connects the two nucleotides together by forming a bond between hydroxyl group attached to carbon number 3 and phosphate group which is attached to carbon number 5 of another nucleotide. So in this case the two nucleotides are connected to each other. Now you can see that the phosphate group attached to carbon number 5 of the nucleotide number 2 is free, and here in this case you can see the hydroxyl group of nucleotide is free. So this is called the 3' end of chain because it contains free hydroxyl, and it can connect, this free hydroxyl can connect to phosphate group of another nucleotide so that it will form a chain. Similarly, here in this case you can see the phosphate group that is free attached to carbon 5, so it is called the 5' end because the phosphate group attached to carbon 5 is free. Now in every chain we always have free phosphate group and free hydroxyl group at another end. Okay, so every chain contains 5' end and 3' end. The end which will contain phosphate group will be 5' end, and the end which contains free hydroxyl group is called 3' end. Okay, so I am sure that you must have an idea about the 3' end and 5' end now. Okay, now in this case you can see the formation of new strand. It's the most important thing to understand, and now look at it. Here in this case this is 5' end. How is this? Why we are calling it 5' end? Because here in this case phosphate group is free, the hydroxyl group of another nucleotide will come and connect to it. So in this case it is a 5' end. Now here you can see it is a 3' end. Why it is 3' end? Because here in this case hydroxyl group is free, and our new nucleotide will come and connect its phosphate group to its hydroxyl group. So in this case a new chain will be formed. Here this is a parental strand which is also called a template strand. This is a new strand which is being synthesized. The new nucleotides are coming, attaching to each other, and this here you can see the nitrogenous base will be attached to this nitrogenous base of the parental molecule. Okay, here you know that guanine will always connect with the cytosine, and adenine will always form a bond, hydrogen bond with thymine. I have told you in my previous lectures, in my previous videos. If you want to know about the chemical structure of the DNA, go and check out my previous videos. Okay, here I have told you about the formation of new strand. How does the new strand will form on the parental or template strand? Now the bases are connected to each other, and the phosphate group and hydroxyl groups are connected to each other to form a phosphate sugar backbone. This is phosphate sugar backbone, and these are nitrogenous bases that are connected to each other by hydrogen bonds. So in this case, a DNA molecule is being synthesized. Okay, now here you can see, you know that the two strands of the DNA molecule are anti-parallel to each other, and this is 5 prime if one chain of nucleotide is starting from 5 prime end, then the other chain will be starting always from the 3 prime end. Okay, you should always know this. Always are anti-parallel to each other. If one strand of the DNA molecule is starting from prime end, then the other strand will always begin from the 3 end, and similarly it will end on 5 prime end, and this chain, the new forming chain, will always end at 3 prime end. Okay, it means that it will contain free hydroxyl group at one end, and it contains free phosphate group at one end. Okay, now I hope that you got it. Let's move toward lagging strand. Now I have told you the new forming strand which elongates away from the replication fork is called as lagging strand. As you know that this point is replication fork. Okay, and now the lagging strand, it is present here. The lower one is the lagging strand. Okay, the lagging strand is always directed away from the, it is moving away from the replication fork. This is here in this case the primase will attach here at this point, because as you know that I have told you that the DNA polymerase will always form a new strand elongated, starting its elongation from 5 prime end to 3 prime end. So here in this case you can see that we have 3 prime, so the new forming chain will always be directed from 5 to 3 point. So this is, here we have we will form up the primer will primase will form a primer here at away from the replication fork and directing away from the replication fork. The primase is formed here, and the new nucleotides are added by the DNA polymerase, and a segment is formed. This segment is called as OKAZAKI fragment. Okay, so here in this case the replication is not continuous, the segments are formed. These segments are called as OKAZAKI fragments, and these are very short segments. Similarly, you can see this is another segment forming here. This is the red color is primer, which is formed by the primase, and after that the DNA polymerase had started its work and formed a new nucleotide chain strand over the old parental strand. So this is called a lagging strand. Here in this case you know that in lagging strand the replication is not continuous. This chain is directed away from the replication fork. Okay, one more thing which is important for you, the OKAZAKI fragments contains about 100 to 200 nucleotides in eukaryotes. Okay, and 1000 to 2000 nucleotides in prokaryotes. Okay, it's an important point for you that the OKAZAKI fragments are about 100 to 200 nucleotides long in eukaryotes and 1000 to 2000 nucleotide long in prokaryotes. Okay, and these OKAZAKI fragments are always formed from 5 prime end to 3 prime end. This is its 5 prime end and it is 3 prime end. Okay, and one more thing which is important for you is that here you can see the gaps that are present between the segments. Now our next enzyme will let you know that how these gaps are being filled. The gaps, ligase. our last enzyme that is used in replication, most important one, is ligase. The function of this ligase is to fill the gaps that are present between the OKAZAKI fragments in the lagging strand. Leading strand does not have the gaps among the nucleotides, but the lagging strands do have gaps between the fragments, OKAZAKI fragments. Here you can see this is one segment and its second segment. There is a gap present between these two segments, and this gap is being filled by the ligase. Okay, now I hope that you got it. Now I have made a summary about all the enzymes and its roles, and every thing you should remember, you should learn. These are solved in this summary. Okay, first is helicase. Its role is to break down the hydrogen bond. Okay, and after breaking down, you know that we need to unclip those two strands of the parents so that they may not connect to each other. So for that we need DNA binding proteins, or SSBP, single stranding binding proteins, that will not allow those strands to coil again. Okay, the third enzyme we have is primase, which will form RNA primer. We know that the RNA primer is very necessary for the replication, because the DNA polymerase 3, which forms the new strand of the DNA, cannot start its work without primer, without RNA primer. The RNA primer is basically a sequence of 10 nucleotides of RNA, which are very important for DNA polymerase 3 for the formation of new strand. Okay, and here in this case, okay, we haven't discussed about the DNA polymerase 1 and DNA polymerase 2 in our full complete topic. Now I will let you know about these two enzymes also. These are also important in the replication of DNA. Okay, DNA polymerase 1, its role is to correct the mistakes in the DNA. Sometimes wrong nucleotides are placed by DNA polymerase 3. It could cause errors. So DNA polymerase 1 will work as a proofreader, which will basically correct all the mistakes present in the new which were maybe created during the formation of new strand. So DNA polymerase 1 will correct all those mistakes. If any wrong nucleotide is placed in new strands, it will replace that nucleotide with a correct one. For example, if the adenine is being connected with the cytosine, so the polymerase 1 will replace that adenine with guanine, so that the correct sequence may be formed. Okay, the second is DNA polymerase 2. We haven't also discussed about this enzyme in our full lecture. Okay, it repairs the DNA. In our life, sometime maybe our DNA maybe damaged, so the DNA polymerase will repair that segment. Okay, and DNA polymerase 3, as you know, that it is the most important and most basic enzyme which will form the new strand of the DNA, and of course it needs the help of primase. Without that, primase cannot start its work. And the final enzyme we have is the ligase. As we know that the lagging strands contains OKAZAKI fragments, and there is a gap between those segments, and all gaps are being filled by this ligase. So it will fill the gaps of new strand. Here I have a short summary about the mechanism of the DNA replication. Firstly, as you know that we have a parental molecule of DNA. First we need to separate both the strands of the DNA molecule. For that we need a helicase. Helicase will separate both the strands. Okay, unwinding of parental molecule, helical strand of the DNA, which is performed by helicase. It will unwind both the strands of the DNA parental molecule, so that we may get template strands. Okay, now on these template strands, new strand will be formed. Before the formation of new strands, as we know that we have to clip the parental strands, and then the primer or primase will come, and it will form a primer, RNA primer, upon which here is the diagram. Let me explain this diagram to you. Okay, firstly we have helicase here. It will break down the hydrogen bonds between the parental molecule. It's the parental molecule, and the parental molecule, the helicase will basically unwind or unzip the parental molecule. Here we have a parental molecule. The helicase will break down the hydrogen bonds present in the present between the nucleotides of the parental molecule. So the two strands will be separated. Here are the two strands. These two strands of the parent molecule will act as a template upon which new strand will form. And here you can see, after the SSBP will will bind to the parental molecule, a primase will form a primer at the end, and the DNA polymerase 3 will start the formation of new strand. Here you can see this is DNA polymerase 3, and this is a leading strand and this is a lagging strand. Here in this case you can see DNA polymerase 3 is working continuously without any pause, without any stoppage at any point. Okay, it will continually form a new strand. It will add nucleotides to the parental strand and will form a new daughter strand, which will elongate from 5 to 3 end, as you know that. Okay, in the lagging strand we have you know that after the primase will put a primer, our DNA polymerase 3 will start its work and forms the segments of the DNA molecule. Here you know that these segments are called as OKAZAKI fragments, and the gaps are remain between these segments, and the ligase will finally fill the gaps between these segments. And this is all about DNA replication. Hopefully you got it, and if you like the video, thumbs up please, and share, subscribe my channel. Okay, Allah Hafiz, see you in my next video.